Good morning to all the teachers and my friends. Today we are going to discuss about the topic sickle cell anemia and thalassemia. Both are inherited blood disorder have a defect in the globin chain of the hemoglobin. So the genetic disease of the hemoglobin is termed as hemoglobinopathy. So this falls into two main groups called structural hemoglobin variants and thalassemia syndromes. So structural hemoglobin variants further classified into HPS, HPC, HPE diseases and thalassemia syndromes, which is further classified into alpha thalassemia and beta thalassemia. So now what is a hemoglobin? Hemoglobin is a protein of RBC which carries oxygen from the lungs throughout the body and carries carbon dioxide from the body to the lungs. So there are four globin chains in the hemoglobin. There are four types of hemoglobin, which is alpha globin, beta globin, gamma globin, and delta globin. These four combine in four different ways to give rise to different forms of hemoglobin, which is present throughout the entire life. So first one is during our fetal stage, HbF, that is fetal hemoglobin, comprised of two alpha globin, two gamma globin, which will be there. Once after the birth, there will be the presence of expression of adult hemoglobin, which is HbA, which comprises of two alpha and two beta subunits, and also a smaller fraction of HbA2, which comprises of two alpha and two delta subunits. So what are all the important genes need for the hemoglobin synthesis? There are two major genes which are involved in the hemoglobin synthesis. One is alpha globin gene and beta globin gene. Now we'll see the both gene locus. The first one is alpha globin gene, which is present in the 16p 13.3, which has a protein of 141 amino acid, has a molecular weight of 115,257 Dalton, and alpha cluster consists of a HS40, which acts as a locus controlling region of LCR, and a zeta, which is seen in the embryonic stage, and two pseudogenes and alpha genes, which is seen in the fetal and adult stage of development. And now the beta globin gene. Now beta globin gene is present in the 11p15.5, which has an amino acid of 147 amino acid and has a molecular weight of 15,867 Dalton. The beta globin gene has a length of 1.6 kb and has 3 exons and 2 introns, and this cluster consists of a locus control region and epsilon, which is seen in the embryonic stage, and gamma, which is seen in the fetal stage, and delta and beta, which is seen in the adult stages. So that's all about the genes. So now we are going to see about the disorder. One is sickle cell anemia and thalassemia. So now we will continue with the sickle cell anemia. Now what is sickle cell anemia? First, sickle cell anemia is one of the group of disorders known as sickle cell disease. Sickle cell anemia is an inherited RBC disorder in which there aren't enough healthy RBC to carry oxygen throughout the body. Sickle cell anemia has a set of conditions called sickle cell crisis and vaso-occlusive crisis and splenic sequestration crisis, where crisis means episodes of pain. Now, a sickle cell crisis is a pain that can begin suddenly and last several hours to several days. It happens when sickled RBC block small blood vessels carry blood to our bones. You might have pain in back, knees, legs, arms, chest, or stomach. The pain can be throbbing, sharp, dull, or stabbing. Now, vaso-occlusive crisis occurs when there is a microcirculation is obstructed by sickled RBC, causing ischemic injury to organs supplied and resultant pain. Last, splenic sequestration crisis, where the blockage of splenic vessels by sickle cells causes sudden painful enlargement of spleen. Due to the complication, spleen have to be removed in an operation called splenectomy. Or in some cases, they will sustain enough damage to the spleen and it becomes shrunken and it will not function, called autosplenectomy. Patients without spleen are higher risk for the infection from bacteria such as Streptococcus, Haemophilus, and Salmonella. So after that, the normal RBCs are biconcave and they are flexible enough to move through the even the smallest blood vessels in our body. But the sickle cell RBC, which have abnormal hemoglobin, which can't pass through the smallest circulation, and hence they are sticky and rigid, which will block the other blood vessels. Hence, therefore, the oxygen capacity will be less, and there will be which leads to a pain, organ and tissue damage. Now, the history of sickle cell anemia traces back to 15th to 16th century. Now, the first descriptive study was done by Dr. James Herring in 1910. Sickle cell anemia was named by Dr. B.R. Mason in 1922. Some of the major sickle cell disease types, mutations, and where they are commonly seen in. Now, HBS, where in the beta globin gene, sixth position, glutamic acid is replaced by valine, which this mutation is commonly seen in the ancestors from Africa, Mediterranean countries, and Arabian Peninsula and India. HPC, when the beta globin gene, sixth position, glutamic acid is replaced by lysine, which is commonly seen in West African descent. Now, the third type is HBE, which in the 26th position, glutamic acid is replaced by lysine, which is commonly seen in Southeast Asia, Cambodia, Indonesia, Malaysia, Thailand, and also in Vietnam. Now, the inheritance patterns is autosomal recessive. That means both the copies should have the mutated genes. So if the offspring, if it has both the normal genes, then it is normal and unaffected. If the offspring has one mutated gene, then it's said to be a carrier or sickle cell trait, where they are also asymptomatic. But when they inherit both the genes, with both the mutated genes, they are affected and they become symptomatic. The main cause of the sickle cell anemia is the mutation in the HBB gene, which is a point mutation in DNA level. The A is replaced by T, which makes a faulty protein. This beta globin gene is present in the chromosome 11, short arm 15.5, produces a hemoglobin B protein. This hemoglobin B protein is a component of the larger component hemoglobin. Hemoglobin consists of 4 globin proteins, that is 2 alpha proteins and 2 beta proteins. This is the structure of normal hemoglobin, which is bound with the heme group. Heme group consists of an ion which will bind with the oxygen. Now, when this hemoglobin enters into the circulation and releases the oxygen, after releasing the oxygen, the abnormal hemoglobin, the beta one, will be in the crystalline form and unable to trap the oxygen molecules. Hence, their oxygen carrying capacity is low. The abnormal form of hemoglobin is called as hemoglobin S. Now, we'll look it into the molecular level. Now, the normal hemoglobin will have the sixth position, glutamic acid, proline, glutamic acid, and glutamic acid. Now, glutamic acid is a hydrophobic in nature. Now, in the DNA, it has a codon called GAG, and in the opposite strand, CTC. Now, the CTC will transcribe into GAG, and GAG codes for the protein glutamic acid. Now, there is a mutation in this A. A is replaced with T. Now, this is GTG as a completely opposite strand, CAC, and it will transcribe into GUG, which produces a protein called valine, and valine is hydrophobic in nature. Due to this, it forms crystalline structure and the RBCs are sickle. This defective hemoglobins will transport oxygen very well, and once it releases the oxygen into the tissues, they form a structure called tactoids, where the beta globin are crystallized and they are sticky ends. This sticky ends of the Beta will adhere to the other HBS molecules and results in the clumping or polymerization, which results in the sickling of RBC, and the sickling of RBC results in the adheres to the endothelial cells and followed by the vasoocclusion, and after vasoocclusion there will be a rapid hemolysis which leads in the decrease in the total number of RBC, a condition called anemia. Now we will look into the symptoms. Sickle cell crisis caused by variety of circumstances like illness, changes in temperature, stress, poor dehydration, and altitude. Types of complication that can result from sickle cell anemia is severe anemia, hand-foot syndrome, splenic sequestration, delayed growth, neurological complications, eye problems, heart disease, lung disease, skin ulcers, priapism, gallstones, sickle cell syndromes. Now we will see every symptoms in detail. Severe anemia. Anemia is the shortage of RBC. Sickle cells are easily broken. The breaking apart of RBC is called chronic hemolysis. Generally, RBC's lifespan is up to 120 days, but sickle cell live for maximum 10 to 20 days. Hand-foot syndrome occurs when RBC block blood vessels in hand or feet. This causes hand and feet to swell, also cause leg ulcer. Sudden hand and feet are often the first sign of sickle cell anemia. As we already saw, the splenic sequestration is the cause of can be two. One is splenectomy or it can be autosplenectomy. But the patients without spleen are higher risk of infection of bacteria such as Streptococcus, Haemophilus and Salmonella species. The delayed growth occurs in the people with sickle cell disease, generally shorter but regain their height by adulthood. Sexual maturation may be delayed, happens because of the sickle cell RBC can't supply enough oxygen and nutrients. Neurological complications like seizures, strokes, even coma can result from sickle cell disease caused by the brain blockages. Immediate treatment should be sought or the condition can be also fatal. Eye problems. Blindness. Blindness is caused by the blockage in vessels supplying oxygen and nutrient to the eyes. Can ultimately damage retina and lead to blindness. Skin ulcers. Skin ulcers in leg can occur if small blood vessels are blocked. Heart diseases. Since sickle cell disease interfere with blood and oxygen supply, it can cause problem which lead to heart attack, heart failure, abnormal heart rhythms. Now, lung disease. Damage to lungs over time related to decreased blood flow can result in high blood pressure in lungs called pulmonary hypertension and scarring of the lungs or pulmonary fibrosis. Lung damage makes it more difficult for the lungs to transfer oxygen into the blood, which result in more frequent sickle cell crisis. Can occur in the patient with sickle chest syndrome. Now, priapism. It is the persistent and painful erection of penis happens when the blood vessels in the penis are blocked. Can lead to impotence if left untreated. The next one is gallstone, one of the complications not caused by a vessel blockage. Instead, they are caused by the breakdown of the RBC. A byproduct of the breakdown is bilirubin. High level of bilirubin leads to gallstones or pigment stones. Sickle cell syndrome. This is the severe type of the sickle cell crisis. Causes are severe chest pain and associated with symptoms such as cough, fever, sputum production, shortness of breath, low blood oxygen levels. Abnormalities observed on chest X-ray can represent either pneumonia or pulmonary infection, that is, death of lung tissue. Also, there is pregnancy complication. Sickle cell anemia can increase the risk of high blood pressure and blood clots during pregnancy. It can also increase the risk of miscarriage, premature death, having low birth weight babies. So we are going to look into the diagnosis of sickle cell anemia. So all the newborns in the US are screened for sickle cell disease. It is estimated that 1 in 12 African Americans will be the sickle cell trait. So the diagnosis can be done prenatally by a process called amniocentesis, and also followed by the routine blood test. In the blood test, we can check for the hemoglobin level. The normal hemoglobin level seen in the males are 13.5 to 17.5 deciliter, and in females 12 to 15.5 deciliter. In the case of sickle cell anemia, blood level will be the blood level will be 5 to 8 deciliter, and in the blood films they can see the sickled RBC, and in the sickle solubility test is also done. The confirmatory test is hemoglobin electrophoresis, where the different types of hemoglobins are appeared. Now the treatment, management, since it is a genetic disorder, it can't be cured. So we can manage by avoiding pain episodes, relieving symptoms, and preventing complications. So the medications and surgical procedures are done to treat the sickle cell anemia. The first drug was used against the sickle cell anemia is azacitidine. When they know that it isn't carcinogenic, then they stopped using that and they used a similar kind of drug which has an activity against sickle cell anemia, which is hydroxyurea, which reduces the frequency of painful crisis and might reduce the need for blood transfusion and hospitalization, which increases the gamma globin, which is required in the which is present in the fetal hemoglobin, that is HbF. Also increases the risk of infection because it will because of the decrease in WBC and platelets. It is advised that pregnant women don't take the drug. Also, drugs like L-glutamine and crizanlizumab are taken. These are FDA approved drugs. Side effects includes nausea, joint pain, back pain, fever, etc. And the surgical procedure includes blood transfusion and stem cell transplant. Now, blood transfusion in an RBC transfusion, RBC are removed from the supply of donated blood, then given through the vein to the person with sickle cell anemia. This increases the normal RBC, which reduces symptoms and complications. Risk factor include the immune response against the patient. Stem cell transplant or bone marrow transplant. Replacing the bone marrow affected by sickle cell anemia with healthy bone marrow from donor. This procedure usually needs a matched donor such as sibling who doesn't have sickle cell anemia. In 1984, there was a first reported sickle cell cure by a stem cell transplant. It was an 8-year-old girl who has a sickle cell anemia with acute leukemia. The bone marrow is taken from his brother, and bone marrow transplant is done, and the girl is relieved from the sickle cell anemia. And the cause of the risk associated with the bone marrow transplant, the procedure is recommended only for children who have significant symptoms and complications for sickle cell anemia. The factors that may cause RBC to sickling are hypoxia, hydration, and acidosis. That conditions can be improved with oxygens and fluids. Immunizations can also prevent the infection for the persons who have low immunity. And children are treated with penicillin against the bacterial infections which may cause sepsis or meningitis. And at last, since it is a single point mutation, gene therapy can be advised, and it is still under research. That's all about the sickle cell anemia, and it's a quick recap. What we have saw: introduction and inheritance pattern, causes, mutation, and symptoms, diagnosis, and treatment. we'll continue with the thalassemia. So let's continue with the thalassemia. Now, what is thalassemia? Thalassemia is a inherited blood disorder in which our body makes an abnormal form of hemoglobin, that is underproduction or no production of the globin chains. This disorder results in the excessive destruction of RBC, which leads to anemia. Now we'll see the thalassemia types. There are two types. One is alpha thalassemia and beta thalassemia. Alpha thalassemia is the defect in the alpha globin gene, and beta thalassemia defect in the beta globin gene. Alpha globin is commonly seen in the ethnicities like Southeast Asia, Middle East, China, African descent, and beta thalassemia is commonly seen in Mediterranean region, China, Asia, and African Americans. Now we'll move on to the alpha thalassemia. Alpha thalassemia is a genetic disorder where there is a deficiency in the production of alpha globin chain or hemoglobin. Alpha chain synthesis is controlled by 4 alpha genes, that is 2 genes on each copy of the chromosome number 16. Now, mostly the mutations are gene deletions. Inheritance pattern is autosomal recessive pattern, that is both the mutated gene is required for the mutation. Now, we'll see the mutation types based on the deletion. Now, first, one defective alpha gene. If one defective alpha gene is there, they are known as silent carrier, where they are asymptomatic because 3 other functional genes are there which will produce enough amount of the alpha gene by which the person is healthy, but they can pass their gene to their offspring too. Now, number 2, 2 defective alpha gene. They are known as alpha thalassemia minor or alpha thalassemia trait, wherein there can be 2 type of deletion over here. One is cis deletion, where the mutated gene is present on the same chromosome, or trans deletion, where the mutated gene is present on the 2 different chromosomes. The cis deletion is most commonly seen in the Asian population, whereas the trans deletion most commonly seen in the African population, and they are also asymptomatic. And third, 3 defective gene which causes moderate disease called hemoglobin HbH disease. For it, when alpha chains are not synthesized in enough amount, there will be the accumulation of the formation of beta tetramer, which causes hypoxia. This hypoxia can cause 2 things. One is damage RBC membrane, which leads to intramedullary hemolysis or extravascular hemolysis, or HbH, which has high affinity for oxygen, thus it does not release oxygen, thus it causes hypoxia. Now, what are all the consequences of hypoxia? Due to hypoxia, it signals to the bone which containing bone marrow, extramedullary tissues such as liver and spleen to increase in the production of RBC, whereas after the production, you can see the enlargement of bones containing bone marrow and hepatosplenomegaly can be seen. If 4 defective alpha gene is there, so that condition is not compatible with life, because no alpha chains or globin chains or products result in the HP barge hydrops fetalis. In the fetal stage, only alpha tetramer is formed. So, or it is also called HB barge. Therefore, no synthesis of alpha globin chain, and this HB barge has high affinity for oxygen, which causes severe hypoxia. Severe hypoxia lead to high output cardiac failure and hepatosplenomegaly, which results in the edema and ultimately results in the hydropsis fetalis, which is not compatible with the life. And if the fetus is not treated, it will be dead in the utero or soon after the birth. Now, beta thalassemia is the genetic disorder where there is a deficiency in the production of beta globin chain of hemoglobin. There is either partial or complete hemoglobin chain deficiency due to the point mutation on chromosome level. Now, what kind of mutations are seen? There are promoter sequence mutations can be seen and splice site mutations can be seen. The inheritance patterns, same autosomal recessive patterns where both copies are required to cause a mutation. Now, if single gene is mutated, beta plus indicates it is reduced production of beta globin gene, and beta 0 indicates the no production of the beta globin gene. If single gene mutation, if only beta plus or beta 0 inherited, then the condition is known as thalassemia minor, where if two mutated genes are inherited for the reduced production, then it goes for beta thalassemia intermedia, or if two genes for no production inherited, it leads to beta thalassemia major. Now, due to this beta globin chain deficiency, there will be alpha globin chain accumulation, which leads to intravascular inclusions, which will damage the RBC membrane. RBC membrane damage may be intravascular or extravascular hemolysis, intravascular hemolysis or extravascular hemolysis. Hemolysis can release the iron and unconjugated bilirubin in the serum plasma. Over time, the excess of iron leads to secondary hemochromatosis, and excess unconjugated bilirubin may cause jaundice. Now, this hypoxia, also hemolysis, will result in hypoxia. Hypoxia, the consequences of hypoxia, which signals to the bone marrow and liver and spleen, which to make the excess RBC, to increase the production of RBC. Thus, you can see the enlargement of bones, liver and spleen. Now, we're coming to the symptoms. Symptoms are, in minor and moderate thalassemia, mostly they are asymptomatic. There won't be any symptoms. In the major thalassemia, symptoms include signs and symptoms of anemia like pallor, shortness of breath, fatigue, and also jaundice, hepatosplenomegaly, skeletal deformities. Complication due to the hemochromatosis is arrhythmias, pericarditis, cirrhosis, hypothyroidism, diabetes mellitus. Other beta thalassemia major symptoms are chipmunk facies, a characteristic facial appearance, and the others, on skull x-ray, hair round end appearance can be seen, or crooked appearance can be seen. Now, how can it be diagnosed? It can be diagnosed by first prenatally by amniocentesis or chronic villi sampling, or in the routine blood test, where you can see the presence of low hemoglobin, low mean corpuscular volume, and low mean corpuscular hemoglobin, and increase in the red blood cell distribution, with that is RDW, where the RBCs are of different size. Now, also you can do peripheral blood smear, which shows the microcytic, that is small RBC, hypochromics, that is pale RBC. Now, target cell or podocytes, which is a bullseye, you can see this kind of appearance. Or in the alpha thalassemia, golf ball like RBC can be seen due to the accumulation of HBS in the RBC. Now, also the lab test indicates the increase in the serum iron, ferritin, transferrin saturation level. Or the confirmatory test for the diagnosis is hemoglobin electrophoresis. Here, where the if the electrophoresis is run based on the hemoglobin weight, you can see the different bands. Now, finally, coming to the treatment, where the mild and moderate thalassemia doesn't require any kind of treatment, whereas severe thalassemia, which require periodic blood transfusion. If you are doing the periodic blood transfusion, there may be the chance of iron overload. To prevent this, iron chelating agent such as a deferoxamine is given, by which it will take the excess iron and remove it by feces or urine. And also bone marrow transplantation can be done, or splenectomy can be done if the spleen is doing extra hemolysis. That's all about the thalassemia. Now, as a quick recap, what we have seen: the first, thalassemia and its types, and alpha thalassemia, and the mutation is gene deletion, and the inheritance patterns is autosomal recessive, and mutation types based on one defective alpha gene, 2 defective alpha gene, 3 defective alpha gene, and 4 defective alpha gene, and the beta thalassemia. Beta thalassemia is due to the defect in the beta globin gene, and the mutation is on a promoter sequence mutations and splice site mutations, and inheritance patterns is autosomal recessive, and there can be 3 conditions, whereas thalassemia minor, beta thalassemia intermediate, and beta thalassemia major. And due to the hemolysis and hypoxia, we can see several symptoms, and at last diagnosed by prenatally by amniocentesis and chronic villi sampling, blood test or peripheral blood test, and followed by the treatment, mainly blood transfusion and bone marrow transplantation can be done. Hope you have understood the video. Thank you.